that I've been leading from the US Geological Survey. Although my name is listed on this presentation alone, there's many people who have assisted with this, and you'll see throughout the presentation that I try to acknowledge my collaborators as much as possible. You'll also notice somewhat of a bias in this presentation towards Lake Michigan, and that's primarily due to both its importance to our partners, but also largely to the fact that it's all in the United States, and we've been able to carry out some of our recent science on Lake Michigan more efficiently than the other lakes because we haven't been able to travel into Canada much lately. So with that, I'll talk to you about how it is we're trying to sort of transform the way that we're measuring and monitoring the status and trends of various ecosystem components to inform decision making in the Great Lakes. Probably it's really no no mystery or no secret to anyone on this call that the Great Lakes are vast and important. I think sometimes it's good to be reminded of just how massive and important they are, however. I would say they're large enough to have sort of a geopolitical importance. Beyond having a large portion of the world's fresh water, there's a massive economic sort of contribution from the Great Lakes states and the Great Lakes portions of Canada to the economies of North America. And a little bit closer to home for the work that I'm conducting, there's a very robust fishing and boating economy that brings in about $7 billion annually. A number of different federal agencies, including US Geological Survey, have roles to play in monitoring and assessing the ecological resources in these lakes. It's basically a massive challenge. When you line the lakes up along the Pacific coast, you can see that much of the exclusive economic zone practically of the Pacific coast of the United States could be occupied by the Great Lakes, north to south. Obviously, they're shallower than that, but there's only a few agencies engaged in the monitoring activity, creating distinct challenges. USGS's mission relative to the monitoring and assessment of resources in the Great Lakes is laid out in our mission to monitor, analyze, and predict the dynamics of complex human and natural earth systems, interactions, and deliver actionable intelligence at scales and timeframes relevant to decision makers. I emphasize the part about scales and timeframes here because the technologies that I'll be talking about today have the potential to radically change the way that we both understand ecosystems, these Great Lakes ecosystems, but also make decisions about how to manage them and mitigate against problems that are occur within these ecosystems. And I also want to sort of briefly touch on the concept of scale in ecology, which is considered to typically have two components. One of those components is the extent, which is essentially the spatial sort of extensiveness of the system being considered, but the other, and essentially, for instance, Saginaw Bay might be the extent at which the state of Michigan is particularly interested in understanding the effects of nutrient effects coming down from the various rivers and watersheds. And that's a natural extent at which the state might work. The federal agencies, of course, are challenged to work across boundaries, across state boundaries. And so the natural extent at which the Great Lakes Science Center and USGS would tend to work is across all 5 Great Lakes. And then the other component of scale is grain, which is what we think of like as the minimum resolution of the data needed to understand the pattern of interest, or within a management context to think about what is the minimum resolution, and that could be temporal or spatial resolution needed to support resource or conservation decision making. Both of those concepts I'll try to sort of revisit towards the end of my talk, but essentially the technologies I'm talking about today have promised to both sort of meet the extent at which the federal agencies are trying to monitor the lakes, but also to drastically increase potentially the temporal and spatial resolution of the information that we provide. And the information that we are bound to provide in a more specific sense, and when I say we in that statement, I'm talking about the Great Lakes Science Center, is we are bound through a memorandum of understanding to the Great Lakes Fishery Commission and the Great Lakes Council of Fisheries, Council of Lake Committees, which is essentially the region's fisheries managers from the states and the provinces and the tribes, to report on annual status and trends of crayfish survey, crayfish abundances. Crayfishes are the little silver fishes that are eaten by the economically important ones like walleye and Chinook salmon in Lake Michigan, lake trout, lake whitefish, etc. So this is all coordinated regionally through the Great Lakes Fishery Commission, and USGS essentially orients a lot of our activities and our science activities to provide information that eventually gets used to support discussions and decisions about limits to harvest, stocking targets for various lakes, and information that would help with restoration of native species and some other topics. The CLC tends to make decisions on a lake-by-lake basis, and so we tend to also organize our science on a lake-wide basis, and they also are making decisions on an annual time frame typically. And so this is also the time frame at which we've historically reported our data. Our data looks something like this, and this is from one of our surveys, which is a bottom trawl survey, where we're trying to estimate temporal trends and the abundances of different prey fish species. The units we usually deal on are kilotons. And we've been running this particular survey for almost 50 years. And it's become sort of an important part of the regional fisheries decision-making context. The way we collect those types of data historically is using large research vessels. And you can see in the upper and right-hand side of this slide, the fleet of 5 different research vessels that we own and operate out of the Great Lakes Science Center, which is headquartered in Ann Arbor. If you look at the map in the middle, you see that we have stations and vessel bases scattered throughout the region. We have one of these large research vessels on each lake. They each have a standing crew, standing captain, year-round federal employees staffing these. And every summer we're out on the lake for months at a time conducting research. However, even with the vast amount of effort that we put into the various surveys that we're doing, we still find that the information we provide can be lacking, and sometimes is in sort of incapable of supporting the needs for detailed information of our partners. So, for example, in a typical annual acoustic survey, which is essentially where we take one of the ships that has echo sounders that are shooting sound into the water and counting the fish that we see and sizing them, we would go out on the lake and collect information like this. This would be overboard sampling using a CTD, which is basically an overboard sensor that measures conductivity, temperature, and water depth, and also chlorophyll, which is it's basically the concentration of pigments, green pigments in algae. Chlorophyll is important because it forms the base of the open water food web. So in a typical annual survey, we would run a bunch of transects. We would drop a sensor overboard and collect a bunch of point samples and vertical profiles of the water column. And in the course of doing those transects, we're also using the down-looking echo sounder to collect these little curtains of data, which I would call an echogram. And these are basically measuring the scattering energy of organisms in the water that bounces back up to the transceiver on the echo sounder. And so you can see typically there's little thin layers towards the top of the water column that represent places where there's aggregations of fish. Where you have hotter colors, you have, more fish essentially. And when you think about this in the context of the extent and volume of the Great Lakes, essentially an annual survey, despite all the blood, sweat, and tears that go into that, months of time by 5 people on each vessel, we're still only surveying a very small fraction of the lake volume. There's some question about the sufficiency of this, especially given recent changes in the ecosystem that have led to increased patchiness of the fish populations, where they're distributed less abundantly and perhaps more patchily. So although we get good spatial coverage, we're not getting particularly high data density. Now, a few things happened in around 2015 that allowed us to sort of dig into this question of sufficiency and start thinking about, are there other tools available that can help us improve the quality of information that we're providing to our partners? And we could conceive of quality as improved accuracy, improved precision, and improved spatial coverage. One of the things that happened in 2015 is that the leadership of USGS headquarters in Arlington, Virginia, really thought that it was time to modernize, and they began to invest in the Great Lakes modernization of the fisheries techniques, and their mantra was better, faster, safer, cheaper. And that's around the time I was hired at the center with some of the funding that was oriented towards that topic. The other thing is that because we're at a time of historically low abundances, which you can see in the bottom chart here, the data that we provide to our partners is basically placed under greater scrutiny. And so, in the past, when there's high abundances, we always pretty much had, people were satisfied with our numbers because there's such high abundances that the fishery was very robust. But now that it's I would say, we're finding lower and lower abundances in our data, it puts sort of fractional differences or errors in our data into under a microscope, and it definitely puts some pressure on us to ensure that both that our effort towards catching the fish is sufficient, and also that we're putting that the biases associated with the methods that we catch are not compromising accuracy too much. And then finally, we got some seed money, and we got some seed money from a place called the National Innovation Center, which is a USGS fund that we applied for some grant money, and we were awarded. And so with my colleague David Warner and a few folks at the Monterey Bay Aquarium Research Institute and BARI, we were able to do an initial test of an autonomous underwater vehicle, which is essentially a torpedo-shaped robot that has sensor payload. We were able to do an initial test using this autonomous underwater vehicle. And in this case, we were interested to document the variation in the chlorophyll A that I was talking about, which is an indicator of lake productivity. And we were also looking for sort of the vertical distribution of chlorophyll and comparing it to other available sources of large-scale data, such as satellites. So on the left here, you can see basically a data product that Barry Lesch published in 2013 that shows the surface distribution of chlorophyll A in micrograms per liter in pixels. And I believe this came from a sensor called MODIS, which is a sensor that has 5 or 6 spectral bands. And so, this is essentially shows where chlorophyll is on the surface of the water. And what we were seeing in our fisheries data around the same time was where we were seeing hot spots shown in red in this diagram. we're seeing hot spots in the abundances of major prey fish species like alewife, which is essentially the preferred food of Chinook salmon. And so we were interested to use this robot to conduct a large-scale survey of the distribution of the basal resource, i.e. the food at the bottom of the food web, at the scale of Lake Michigan. And we did that. Over 19 days, we conducted two, I'll call them mega transects for lack of a better word, from Muskegon to Holland in the first case, and from Pentwater to Charlevoix. And you can see this is the light availability data on the right. And each time you see white, that's the daytime observations during the survey. Throughout the survey, we had the AUV, the autonomous underwater vehicle, doing vertical profiles of the water, where it's basically yo-yoing up and down and measuring a number of characteristics, including chlorophyll A and temperature and some other things. You can see that we were able to map a very in great detail the vertical patterns in chlorophyll distributions, to which all the other species in the open water food web are essentially responding. Plankton are feeding on these, larger plankton are feeding on those plankton, prey fish are feeding on the larger plankton, etc. What we found was that we got a great degree of detail about the distributions of our interest, our topic of interest, chlorophyll. We saw consistent distributions of deep chlorophyll, of a deep chlorophyll layer, which on the horizontal axis here, we're looking at increasing chlorophyll in each plot versus depth. And so, and we use this to essentially look at those satellite-based products and say, how sufficient, how representative is the satellite-based estimate of the productive capacity of the lake relative to the entire water column? And so what we found was that essentially that surface chlorophyll is not indicative of the abundances of that deep water chlorophyll, and which is essentially justifies the continued use of instruments like the autonomous underwater vehicle to conduct in situ remote sensing of these important patterns. And when we compare the data density from this, you can really see a stark difference. We went from maybe 3,000 point observations, if you assume we're taking a measurement a second on each of these vertical casts, to upwards to 140,000 data points, and across these continuous data curtains. And in these data curtains, you can see the deep chlorophyll reflected in August in Lake Michigan in 2016. So what this led us to sort of postulate, and what it also confirmed, is that advanced survey technologies are currently now ready and capable to do full-scale surveys of the world's largest freshwater seas. And we may be in a position to evolve from our status quo, which is essentially vessel-based sampling with some acoustic sensing and trawling. And also we have a nice acoustic telemetry program linked up with the Great Lakes Acoustic Telemetry Observing System, GLaDOS. But we're thinking about how can we evolve our program to incorporate new technologies and new approaches that have a high technical readiness to detect other patterns in the lake environment. And the set of patterns that we're interested in are what I'll be talking about today in two little vignettes. Essentially, in this diagram, there's a suite of technologies, including aerial remote sensing, computer vision with autonomous underwater vehicles that are flying near the lake bed, autonomous surface vehicles. In this case, this is called a wave glider. There's also diesel-powered ones that are like 15 feet long that you can remotely helm from shore. And we're also looking into the use of environmental DNA, which is essentially is DNA that you filter out of the water column and use to discriminate the presence and or abundance of species, as well as other characteristics about the biological community. So today I'll give you two vignettes related to these topics, starting with the one using autonomous surface vehicles and a little bit of a mention of DNA here. So this is a project that is funded with new appropriated money that came to USGS through a new law called the Great Lakes Fisheries Research Authorization Act. We received a $1 million increase in our operating budget, and with the Council of Lake Committees and the Great Lakes Fishery Commission, we focused the use of those funds on two specific shortcomings of our current survey work at USGS. And one of those is focused on acoustic fish abundance estimates, which again is these sort of down-looking echo sounders. And this is a massive collaboration. These are the collaborators that assisted on Lake Michigan, including a bunch of folks from USGS, Fish and Wildlife Service, Michigan DNR, Cornell, Michigan Tech, Monterey Bay. And so essentially, the background here is that what I'll call systematic biases are lowering the accuracy of our acoustic surveys. So a systematic bias is essentially a characteristic of a survey method that's inherent to a survey method that degrades the accuracy and/or precision of your data. In the case of vessel deployed echo sounders for fish counting, there's three major sources. One of them is the noise of the vessel itself is known to cause the fish to flee before the vessel. In some cases, there's been shown to be a 60% reduction in the density of fish by noise alone. There's also what we call these dead zones. There's an acoustic dead zone at the surface, which is basically an area where we cannot detect fish because the transducer, that basically the echo sounders deployed below the water surface to begin with. There's a very low beam volume, and the vessel itself is scaring fish away from the sensing area. And then there's also this bottom acoustic dead zone, which is caused by the fact that the beam spreads out and it hits, the first thing it hits is usually, well, the hardest thing it hits ends up dominating the signal. So if there's any variability, vertical relief in the lake bed, what we see at the very close to the lake bed is we see the lake bed itself instead of any fish targets. So we've gone about devising a study to quantify bias using advanced survey technologies. Now, what is bias in a specific sense relative to this problem? This paper by Vaba et al. Basically, what they did was they put a fisheries echo sounder on a buoy. It was pointing down, and there's no other vessel noise in the area. So essentially, you have a quiet source of information about fish densities. Then they ran a noise-producing survey vessel right past the buoy over and over again. And what they saw is this is the ping number. So this is essentially how many pings of the echo sounder leading up to when they passed the buoy here and then past that. What they saw is that, and the baseline for the buoy is the top line here. What they saw is that there, as the ship approached the buoy, there was a little increase in fish densities, which could have been from the flight of the fish in front of the vessel. And then they saw a scattering the fish densities away from the buoy location until basically a minimum, a local minimum. And then there's a slight recovery in densities after the vessel passed. So for us, bias is the difference between the reference biomass and the ship biomass. In this case, this isn't actually a measure of fish biomass. This is more of a measure of the scattering energy that's bouncing back off the fish, but it's an accurate proxy for density and biomass. So what we're trying to do is collect these sort of phase one data, and we're also trying to understand for the Great Lakes fishes specifically when the fish start to flee the vessel and other questions like that. So just the study objectives essentially are to quantify vessel noise effects, quantify the acoustic dead zone effects, but ultimately what we're trying to do is develop models to correct for the bias. So we're essentially going to use that measure of bias that I just showed you as a response variable in a model to essentially try to both correct our future vessel-based data as well as backcast our historical data to more accurate numbers of density. And we're, parenthetically, we're also evaluating new technologies that could potentially contribute to our surveys going forward. One of those technologies is called sail drone. It's a wind-powered, non-motorized, uncrewed surface vehicle. It carries Fisheries echo sounders identical to the vessel-based ones, and operates silently except for a little bit of noise from the hull slapping the water. It can stay out for up to 24 months if there were no ice. It has high endurance, and it's basically controlled from shore in California, actually. We submit routes to Saildrone. They submit it to the vehicle. And this thing does not move very quickly. It's not a big navigational hazard, but we did issue navigational warnings leading up to this. The other technology that we're using is the same autonomous underwater vehicle that we used for the chlorophyll study, except this time we've outfitted it with echo sounders. So these red hockey puck looking things are up and down looking echo sounders for counting fish. This operates very quietly, and it also has a high range, as you observed earlier. In 2021, we ran about 3,600 nautical miles with 2 drones, starting in Holland and going all the way around through the straits into Lake Huron, a little bit northeast of where you all are sitting. And at the same time, we had 3 different research vessels on the water conducting overtakes to get comparative data. We ended up with about 40 different 61 overtakes at about 40 locations. Oh, I'm sorry, 61 overtakes at many locations. And we had another 40 midwater trawls to support the understanding of the data. And then we're also conducting noise profiling of all the vessels to incorporate in our modeling. So here's a picture on the bottom left of the vessel overtaking the Saildrone in the middle of Lake Huron. This is a picture I took myself. So we don't have the data yet to show you, but we will in the relatively near future be able to present these out to people. But this represents an exciting advance in our science. The other thing that we did concurrently in the southern basin of Lake Michigan, which I would define as sort of the region south of Muskegon and Milwaukee, is that we were running a second payload with the Monterey Bay Aquarium's autonomous underwater vehicle, which is a payload that takes filters of water onto very fine filters that are fine enough to collect eDNA from the water. So we're essentially collecting DNA from organisms that have shed slime, or they've defecated, or essentially, they've essentially left a trace of themselves in the water. And those are collected on filters within these little cartridges and preserved until the robot gets back to shore. So we launched from Holland and retrieved in Holland. You can see each one of these dots in the lower left are places where we collected an eDNA sample. These tended to be between 1 and 2 liters, and we have a known volume for each sample. And you can see that these weren't just at the same part of the water column, they were in each of the different density layers, including the epilimnion, the metalimnion, the hypolimnion, and near the bottom. The lake stratifies in the summer, and the stratification causes density differences that prevent the exchange of water between the different density layers of the lake. So we're using these data to compare measures of prey fish relative abundance, which is essentially the proportion of fish A, B, C, or D that was present in a sample. And we're essentially testing whether or not the eDNA is a suitable proxy for a trawl-based catch, which is our historical sampling method on the large vessel. If the answer is yes, the eDNA approximates the trawl catch, then it puts us in a position to conduct fully autonomous surveys without any crewed vessel at all. We could send a Saildrone out with an AUV and essentially have them sample in coordination with one another and conduct the entire fisheries survey autonomously. That's not necessarily something we are moving towards very quickly, but this is more of a research and development effort and testing the possibilities with these technologies. Moving forward here, the other vignette that I'd like to talk to you about is one where we're coupling, we're largely, what we've done is developed an in situ remote sensing capability with a autonomous underwater vehicle that we've modified with cameras. And in some cases, we're coupling that with aerial remote sensing data collected with LiDAR systems, which essentially give you elevation data about the lake bed in the nearshore environment where the water is clear enough. This is a project that I'm working on with a senior biologist at our center, Chuck Medengen, and some others, including Chris Rusi from Michigan Tech. So the purpose of this survey is to improve our estimates of a fish called round goby, which you can see in the upper left here. Round goby live in rocks and they like the shallow water areas in the summer, and those are two habitats that we don't sample well with our large vessels. And so for a long time, the fisheries community has suspected that round goby have been far more abundant than our surveys have been saying. And the purpose of this project is to essentially evaluate whether that suspicion is true or not. Now, round goby have very strong affiliations to substrate, the type of rock or material on the bottom, silt, sand, pebbles, boulders, cobbles, etc. They also tend to occur in areas that have filamentous algae called Cladophora. And they also consume dreissenid mussels, which are the quagga and zebra mussels that make up the majority of benthic biomass on the bottom of the lake. And so rather than seeing this just as an effort to count gobies, we've sort of attacked this as if it was an effort to understand a four-part system wherein habitat dictates the patterns of these three other endpoints and where there's strong interactions among each of those. Why are round goby important? They're important because they make up a huge portion of the diets of fish like lake trout. They also make up the diets of walleye, yellow perch, lake whitefish, and these are all species that, if you've lived in the Great Lakes for a while, you know are important prey fish species. And there's commercial fisheries and tribal fisheries oriented around these things. So essentially, what we're trying to do here is develop a new technology and a new way to assess them. And the way we're doing that is using what I call GobyBot. Sometimes we also call this robot Bubbles. But this is about a 100-pound robot. It's 9 feet long. Notably, it goes to 600 feet or more depth. It carries a 9-megapixel color camera here and a grayscale stereo camera. And the stereo camera gives us a sort of high-resolution 3-dimensional picture of the lake bed. This takes 5 frames a second along track. And so we're collecting continuous photo transects with this. And it has about a 15-kilometer range on a single battery charge. The way we use this is we take a raw, the other piece of information is that there's a 10-inch hull section in the front of this. That's a very sophisticated gyroscope that can detect inertial changes due to acceleration. That could be due to density differences in the lake. It could be due to currents, upwelling. And so this robot can compensate for that and also estimate its position underwater, meaning latitude and longitude underwater. And so each of the photos has a latitude and longitude. They're geotagged. Our process is to label those fishes. So if you look here closely, you can see some fish in this picture. We detect them and label them. We're moving towards machine automated solution for that, but right now we've been doing that with humans. And while we're working on the machine learning solutions. And then once you have the fishes traced, you can essentially size them. And once you have their length, you can estimate their weight using standard curves. And we can also estimate the area of the photo because the camera's looking down and there's no sort of profile perspective. So because it's looking down and we know our altitude, we can essentially make a pixel size estimation, which is how we're attributing length and area to each photo. So it gives us the ability to estimate density, either numeric density or biomass density, which is measured as grams per meter squared or kilograms per hectare, something like that. And eventually what we're trying to do is extrapolate lake-wide biomass. And so in 2020 and 2021, we conducted lake-wide surveys. This shows you the data. These are all the usable data that we collected in Lake Michigan in July and August 2020. This represents about 56,000 images. There's no overlapping images. We subsampled them, so there's no overlap. Our total sampling effort that summer was 300 kilometers. And out of those, we labeled almost 40,000 individual fishes and therefore are able to size them and weigh them. And so by comparison, our bottom trawl survey every year gets about 6,500 fishes. And so this is a much more efficient way to quote unquote catch a fish in the water. The types of data we get from this are total length versus frequency, histograms, which we get from sort of fitting this red line down the middle of each fish's body and then attributing its length and summarizing it. And it gives us the ability to look in different depth zones, 0 to 5 and 5 to 10 meters, for instance, vary from these deeper depths because you see a bimodal distribution where you have two peaks. And so you have a peak in small individuals evident in the 0 to 10 meter data that's not apparent in the deeper water data, suggesting that the young fish are living in the shallows every year. And so when we do the biomass extrapolation, it's essentially a big exercise in arithmetic. You have depth bin specific fish density, you have the area in each depth bin, and you end up with a kiloton estimate when you do a unit conversion. And so for Lake Michigan in 2020, we just based on area, we would say there's 10.2 kilotons of round goby, which is something like, I don't know, 850 dump trucks full. However, we know that round goby like hard substrate, they like rocks. And so what we did was we used our imagery from our AUV to estimate fine and coarse substrates using a machine learning process. And so instead of just doing an area-based extrapolation, what we're doing now is an area and substrate extrapolation. And so here's showing the densities in different substrates within depth bins. And in fact, it supports the statement I made that you have a higher density of small individuals in fine substrates. And so, our substrate models are pretty effective. We're using both the stereo data and the color data. I think the bottom line here is that we're predicting greater than 95. We're predicting these substrate classes with greater than 95% accuracy on a large data set. And what we've tried to do to enable the extrapolation is we've tried to extend the local scale AUV image-based substrate predictions out to the landscape scale. And this is where the satellite imagery or the aerial photos come in. I'm sorry, the aerial LiDAR data. So if you go to the lake level view, if you search on Google, NOAA lake level viewer, you'll find a great data set that for every 3 by 3 meter pixel in the 0 to 20 meter zone of each lake, except in the areas that have high turbidity, you'll find information about elevation. And so what we're doing is we're mining that information for predictors that summarize the texture of the lake surface. We're linking it up with our substrate predictions, and we're coming up with a coarse versus fine estimate at the scale of the entire bathtub ring of Lake Michigan. And these models are also working really nicely. And you can see sort of some of the pattern here where we're projecting the results of coarse versus fine onto a 3 by 3 meter raster layer. And so again, doing the arithmetic here, where you have fine, this is the same calculation as earlier, except we have area broken down by fine and coarse, and we're using the predicted coarse versus fine substrate from this slide to sort of fill in the proportional representation within each area bin. Essentially, what we get at the end of that is slightly lower number, close to 9.5 kilotons of fish, 850 dump trucks minus 40 or so. And I think notably, this number makes this fish the most abundant fish in Lake Michigan. It used to be bloater, which is a whitefish relative. So, as I mentioned, we're working towards automation, and we're finding that even in slightly degraded images like this one, we're able to put these bounding boxes around fishes that are present in the image. So we're using the hand labeled data to train machine algorithms using an approach called deep convolutional neural networks, which is sort of a fancy AI-based pattern recognition approach. And we're also working on actually having the computer trace the outlines of the fish, which you can see here in these little panels here. And we're seeing very good results on well-resolved high-resolution GoPro data that are a little higher quality than our AUV data. So we're not quite there with our robot to do this sort of accuracy, 97% assignment of the edge of the fish, but this is where we're moving. So to start wrapping this up a little bit, what we've been able to do is demonstrate the feasibility of advanced survey technologies for informing Great Lakes resource management problems. We've essentially concluded that the high readiness technologies are well scaled to the extent of the Great Lakes, meaning that we can monitor the lakes at scale with at a large spatial extent and a high granularity. In other words, a high resolution. Each of those images that I was showing you has each pixel is about 1 millimeter by 1 millimeter. What we've also shown is that the big data analysis that goes on in the background to sort of crunch all these data up, manage hundreds of thousands or millions of pictures, is also sufficiently advanced to use it in routine monitoring. And so the future that I see is one where the hardware, i.e. the robots, continue to advance. The software, which is automated routines and data pipelines, also advance. And so I see the future as one where there's multiple mobile assets deployed on each lake. Those will probably become self-charging and they'll stay in the water for entire summer season and not have to get taken out and recharged. And eventually the hope is that essentially they're just reporting their data up to the cloud, and we can begin to work on an informatics pipeline, which is not dissimilar from the way that USGS currently manages the Landsat data program, which is the longest running color satellite photography effort that has absolutely transformed the way resource management takes place. So, in order to accomplish all these things, we really need to coordinate. And when I say we there, I think it's a process where we coordinate across sectors, across state boundaries, across tribal and federal boundaries to kind of come up with a well-organized strategy. And we target the endpoints of the data collection to very specific user needs. There's a good deal of theory on sort of the best ways to do that. One of those is using an approach called collective impact, where you essentially organize your partners to solve complex problems. And I think we're already starting to do this, at least within the federal family. So with that, I'd like to thank you for your attention. There's a whole number of folks. This has not been an inexpensive project. And it's also not been one that I've done by myself. And so I'm really just a mouthpiece for a large collaborative. So with that, I'd like to thank you and take any questions.
Thank you, Peter. Thank you for giving us this talk. We want to allow the members here who in the talk to put questions in the chat box, or if you want to raise your hand to ask some questions. I can start it off. I saw all this technology. I was wondering if it can be also used for ocean or sea scenarios.
Absolutely. Yeah, I mean, most of these technologies were essentially taken from, they're developed in a salty coast, saltwater environment and brought to the Great Lakes. And so the majority of sail drones effort, for instance, has gone out all over the world in the oceans. In fact, this is the first time sail drones operated in the Great Lakes. And similarly for the autonomous underwater vehicles, Monterey Bay Aquarium Research Institute is located on Monterey Bay, which has this, deep submarine canyon that goes to something like 3,000 meters. And that's where they've been, initially they were funded by the Packard Foundation just to do ocean R&D development. And so, yeah, it's no mistake that those things come out of the oceans, but the Great Lakes are large enough water masses that we get oceanic dynamics, which is why I think they're more appropriately called, inland seas than lakes.
Yeah, there's a few questions in the chat. Someone said from Limnotech that you must be drowning in data, and then another one said, could you use volunteers? So I think those two things might go well, because you said you have to go over every single image and annotate what's a fish. So this person asks, is there a way, Renee asks, is there a way you could use students or citizen scientists to help you with this data?
Yeah, so I'll comment quickly on John Bratton's statement first. We're not drowning. We have a large investment in data storage infrastructure and computation, and so the way we're approaching that, John, is by moving our data sets to the same location that the Landsat satellite image program is managed, which is the Earth Resources Observing System, EROS, in South Dakota, which is a USGS supported resource. And so we're essentially using their high performance computing capabilities to try to deliver these surveys. And then in terms of, so right now volunteering is tough because, well, firstly, COVID has essentially put us down to like skeleton crews only. So we don't have a great way to engage volunteers right now because it's really USGS personnel only. However, I think that's going to loosen up. And one thing that has been successfully accomplished by others, namely EPA, is they've used what would you call it? It's basically a crowdsourced image labeling platform called Zooniverse. And we don't have our data up there yet, but I think it's a great idea. And what EPA did was essentially they said, they asked citizenry in the public to say, to look at video data they had collected and say, is this, are there mussels or round goby in these pictures? And they essentially put together a large data set using that. And so I think there's some possibility of that. Because we've been sort of wrangling our data so much, we haven't yet gone to the step of setting that up. But I think that's definitely possible in the future, and I'll certainly be beating the bushes for volunteers when the time comes. So the next question is, could you comment on typical time periods for collection of data? Could the new technology extend sampling periods? Is there any data addressing climate change and changes in ice coverage that may have an impact on lake biomass? So I think the answer to all of those is yes. So the typical period for a sail drone could be the entire ice off period. That could be from April to November on most lakes. And one advantage of the technologies is that you don't have to stop and sleep. These technologies just go. And the other advantage is that at the shoulders of the season, when the weather gets really bad, there's really a very real risk to the crews and the vessels that go out there, and a risk of injury or death. And these technologies, essentially could help us get better observations at the shoulders of the season, but also even under ice. And then, the sail drone's collecting every 15 seconds or something. It's collecting a temperature observation. The Tethys vehicle is collecting information about the lake stratification and temperature and dissolved oxygen and other characteristics. So I think there's great potential to establish a trend data set where we have a time series of observations that are relevant to climate change prediction and understanding changes and the drivers of ice cover change. But we haven't yet put the pieces together to link that to fish biomass or anything like that. It's not to say it's not happening. There's some efforts within my center to make those associations. And I think what it would require is an annual survey that you can count on to establish a time series of data. And then on David's question, what is being done? So, perhaps that that gives a little bit of an indication. I mean, 1 year of sail drone data or a couple of years of autonomous underwater vehicle data aren't sufficient replication to make a lot of strong conclusions about climate change effects. I think that what we need is to draw those out over time. I think what we also need is to use those in situ technologies as ground truth systems for sophisticated generation of models where they're modeling the currents, they're modeling temperature, and to validate those and bring those models up to the point where we can essentially forecast ice cover, and we can forecast ice on and ice off and currents under ice and when there's no ice, because those are the sorts of things that would essentially allow us to sort of begin to put sort of a clear set of knowledge, a body of knowledge together around climate change in the lakes and its effects. So could the eDNA be used to assess diversity and the presence and biomass of invasive species? So the answer is yes. Currently we're dealing with relatively small samples of water. 2 liters is not a lot of water. There's a lot of molecules in that water of DNA in many cases. And so, and we're actually entraining whole organisms in our filter as well. So if there was an invasive zooplankton, we should be able to detect that with metabarcoding and, next generation sequencing type approaches. I would say that that the future of eDNA, at least from, it's not my specialty, but from the people I talk to who are specialists, is to take large volume samples and to use sources of molecules that have sources of DNA that have a much higher information density. And specifically, that's the nuclear DNA that's held in whole cells. And with nuclear DNA, you can come up with a pretty sophisticated set of markers to determine species, A through Z, through AA, et cetera. And that would require large volume filtering and sophisticated genetics, but that stuff's it's all at our fingertips. It's all doable, I would say. What models do you currently feed data into, if any? Is there a plan to increase data assimilation going forward? So we're using a mix of traditional machine learning methods like random forests, and then deep learning methods like convolutional neural networks, like UNets and RetinaNet. So RetinaNet is like the object identifier algorithm that we're using. And essentially what those require is a lot of training data where you have the features of interest annotated. You feed that into the computer numerous times, you hold some out for validation. And generally speaking, those techniques have been working well for us. In terms of data assimilation, I'm not exactly sure what you mean by that, but so Shelby, if you could please, type another comment. But what I will say while you're doing that is that what we're trying to do is move the computation from the lab where we have local PCs or we have a high performance computer at Eros working on the problem. We're trying to move it from the lab to what I would call the edge, which is to say that we're trying to move the calculations from the laboratory to the AUV itself. And we're not going to train our models on the AUV, but what we would do is we would take a trained model, we would have it on a second CPU on board the AUV, which there's 2 on board right now. And there's some, there's enough RAM and there's enough processing power to essentially just start cranking through our data. So we could do substrate assignments, we could do density summarization, perhaps. We could do other things like cladophora, algae, percent cover, and whatever the solution is that our machine learning process yields. So that's why I think the future is one where the assets just stay in the lake. They'll do all the computation themselves. In 5 years, a little cigarette box size computer is going to be able to do massive computation. And so that's the future that I hope to see within my career. Adrian, looking at the time, I'm going to go ahead and maybe send it back to you and make sure you don't have any closing comments that you want to deliver or anything like that.
Yeah, thank you, Dr. Esselman. I think if you want to continue asking him questions, his email is on the ad, and we'll have it up on the Water at Wayne and the Healthy Urban Waters web link. So you can certainly keep asking Dr. Esselman questions on this interesting topic that he shared with us. I think it's very important. Like I was remembering Jacques Cousteau said, go and see it, right? So we need to see what's down there in the water to make better decisions going forward. So thank you again, Dr. Esselman. And remember, everyone, we have our next Water at Wayne on March 31st. And thank you again, Dr. Esselman, for taking the time to speak to us today.
My pleasure. Thanks so much for having me, Adrian. It's always a pleasure. And I guess there's 1 minute here. John Bratton asks about increasing sensor payloads on fish themselves. It's a great idea. I think that the tagging that's going on, the telemetry, I would say the telemetry network, GLaDOS, is probably the gateway to that sort of thing, because they're really interacting with a lot of individual fish in every year. And the tags themselves that they're implanting are getting more and more sophisticated. One example of that is that there's some tags now that can essentially tell you if a fish has been eaten by another fish, because the stomach acids in the predator end up like burning through the telemetry tag that was in the body of the thing that it ate. And that's still pinging out of the body of the predator, at least until it eliminates it from its system. And so that's sort of an example of maybe a direction that's interesting. And I haven't given it tons of thought though, John, and I'd love to hear your ideas on it, because I know you're no stranger to these things. So Adrian, thanks again. I appreciate everyone, and I look forward to seeing you out and about.
Yes. All right.
Bye-bye.